Hello and welcome to my channel Sam Biology Class. Friends, today we will discuss about RNA interference. Before that, I request all of my friends, please subscribe my YouTube channel and also watch, like and share my videos with your friends. RNA interference is an RNA dependent gene silencing process. It naturally takes place in nearly all eukaryotic organisms where it provides defense against viruses and transposons. It also regulates gene expression. RNA interference, abbreviated as RNAi, was discovered by Andrew Fire and Craig Mello. Was discovered by Andrew Fire and Craig Mello in the nematode worm Sinorhabditis elegans. Sinorhabditis elegans. Sinorhabditis elegans. They introduced short segments of double-stranded RNA into the cells of Sinorhabditis elegans, which leads to the degradation of messenger RNAs. For their work in C. elegans, Fire and Mello were awarded Nobel Prize in Physiology or Medicine in 2006. RNA interference is initiated by short double-stranded RNA molecules in the cytoplasm. The initiating RNA can be exogenous or endogenous. Can be exogenous or endogenous. Exogenous double-stranded RNAs are derived from outside, for example, from infection by virus with RNA genome, from laboratory manipulation, and from other sources. Endogenous RNA, on the other hand, originate in the cell. This includes microRNA, which is transcribed from RNA coding genes in the nucleus. RNA interference functions by using small double-stranded RNA molecules to silence specific genes. These RNA molecules either cleave the mRNA or prevent its translation into proteins. Important components of RNA interference are short hairpin RNA or shRNA. Number 2, small interfering RNA. Number 3, microRNA. Number 4, dicer. Number 5, DROSA. Number 6, RISC. RISC stands for RNA-induced silencing complex. Out of these components, dicer is a ribonuclease 3 enzyme. It functions in the cytoplasm where it cleaves free microRNA into microRNA. Another component is DROSA. DROSA is also a ribonuclease 3 enzyme but it functions in the nucleus. DROSA is a part of the microprocessor complex. This figure illustrates the mechanism of RNA interference. As already said, RNA interference is initiated by two types of RNAs. First one is exogenous RNA and second one is endogenous RNA. Exogenous RNAs are derived from outside, for example, from virus infection. Exogenous double-stranded RNA molecules are imported directly into the cytoplasm. These molecules initiate interference by activating the ribonuclease 3 enzyme dicer. Dicer is a ribonuclease 3 which cleave short hairpin RNAs in humans and double-stranded RNA molecules in plants to produce small double-stranded fragment of 20 to 25 base pairs with the two nucleotides overhang at the 3' end. These are the two nucleotides overhang which are present at the 3' end. The small double-stranded RNA fragments are called small interfering RNA. In addition, RNA interference is also initiated by endogenous RNA. Endogenous RNAs originate in the cell, for example, microRNA. MicroRNA is transcribed from RNA coding genes as a long precursor called primary microRNA. This hairpin precursor, called as primary microRNA, is processed into pre-microRNA by DROSA, which works with the protein DGCR8. Pre-microRNA is then transported to the cytoplasm. In the cytoplasm, pre-microRNA is cleaved by dicer to produce double-stranded microRNA. So friends, when exogenous RNAs are cleaved in the cytoplasm by dicer, small interfering RNAs are produced. On the other hand, microRNAs are obtained from the cleavage and processing of endogenous RNAs. Small interfering RNAs or siRNAs are short double-stranded fragments which are then separated into single strands. Out of two strands, only one strand, known as the guide or antisense strand, is incorporated into an active RISC by RISC-loading complex RLC. The other strand, known as the passenger or sense strand, is ejected. RISC-loading complex includes dicer2 and R2D2. This complex is crucial to unite AGO2 and RISC. Now the question is, what is RISC? RISC is a multi-protein complex whose complete structure is still uncertain. This complex functions in gene silencing via a variety of pathways at the transcriptional and translational level. One of the key components of RISC, that is AGO2 or ARGONOT2, is a protein that acts as an endoribonuclease which cleaves specific mRNA transcripts guided by small interfering RNA and microRNAs. After incorporation into the silencing complex, the guide strand is used as a template to recognize complementary mRNA transcript. Once found, the transcript is cleaved by the endoribonuclease activity of ARGONOT2. So in this case, the guide strand, which is now a part of the silencing complex, base pairs with specific mRNA, which is then cleaved by ARGONOT2. So in this case, there is destructive cleavage of mRNA transcripts. Like small interfering RNA, one strand of the microRNA is incorporated into the silencing complex. But unlike siRNAs, microRNA loaded silencing complex scans cytoplasmic mRNAs for potential complementarity. Instead of destructive cleavage by Argonaute-2, microRNAs rather target 3' untranslated regions where typically they bind with imperfect complementarity, thus blocking the access of ribosomes for translation. The 3' untranslated region of mRNAs contains binding sites for microRNAs as well as for regulatory proteins. By binding to specific sites in the 3' untranslated region, microRNA decrease the gene expression of various mRNAs by either inhibiting its translation or by causing direct degradation of the mRNA transcript. On one hand, small interfering RNA base pairs perfectly with the transcript that leads to mRNA cleavage in a single specific target. On the other hand, microRNAs, because of their incomplete base pairing, can bind with different mRNAs of similar sequences and inhibit their translation. In this way, by using double-stranded RNA molecules, specific genes can be silenced at the post-transcriptional level by RNA interference. Friends, this is all about RNA interference. For more updates, subscribe my YouTube channel, Sam Biology Class.